Since its initial outbreak in the Chinese province of Wuhan, coronavirus, also known as COVID-19, has spread globally, becoming a pandemic that still at the time of writing has no vaccine nor protection beyond preventative and diagnostic measures for disease control. To aid in COVID-19 identification, many laboratories are researching alternative tools for wide distribution without the need for currently used PCR machines. For instance, Long et al. Had discovered that the seroconversion of COVID-19 antibodies, as shown in Figure 1, can quantify and determine the extent of patient immune response, enabling effective pathology prognosis. Individually, viruses are defined as non-functional parasites nanometers in size. However, once specific cellular substances are present, they exponentially replicate themselves by hijacking and subsequently reprogramming their host for duplication. Being encoded in either DNA or RNA format alongside a double or single-stranded nucleic acid, all virions, defined as an entire virus, are encapsulated in a protein shell represented in Figure 2, and can range between a limited 4-protein encode up to the most Deladinian 200. Viruses pose a unique risk towards those susceptible, as unlike other pathogenic material, such as bacteria or parasites, its substantial potential for propagation and mutation can be attributed as an evolutionary response to selective environmental pressures. Virions account for more than 2/3 of all recent pathogens infectious to humans and can have vast pathologies, ranging from the mild yearly flu, commonly known as seasonal influenza, to more deadly, genetically distinct variants such as the H1N1 virus, historically noted as the Spanish flu, which in 1918 was classified as a pandemic, having killed over 50 million people. Mutation rates are highest in single-stranded RNA-encoded viruses, with a negative correlation between genome size and rate of mutation. Predominantly, RNA viruses cause more errors due to its fundamental replication process, as unlike DNA virions, which take advantage of its host polymerases for copying, encode their own replication machinery, creating optimized mutation rates that directly affect their environmental fitness, with offspring up to 1 to 2 mutations different. Classified as a pandemic by the World Health Organization in March 2020, COVID-19, a single-stranded RNA virus, has already been sequenced in labs globally, identifying 80 genetically different genomes. With future vaccine efficacy of considerable concern due to the aforementioned mutated potential, prevention methods and diagnostic tools are currently the only line of defense against the ongoing pandemic. Genetically similar to the coronaviruses severe acute respiratory syndrome and Middle East respiratory syndrome, SARS and MERS respectively, SARS-CoV-2, more commonly known as COVID-19, is a recent variation belonging to the Orthocoronavirinae subfamily originating from the Wuhan province in China. Much like its family, COVID-19 is a single-stranded RNA virus that can infect and instigate a pathogenic response in both animals and humans, with the latter linked to lethal complications of the respiratory system. Genetic sequencing has revealed the virus was most likely acquired as a zoonotic infection, postulated to be concurrent with certain ethnic lifestyles involving the consumption of wild and exotic animal meat. However, Kah Wai et al. Noted that the exact species the virus is spread from is still under investigation. Additionally, the demographics at greater risk, such as the 60-plus age bracket, obese individuals, and those with chronic respiratory diseases, most notably brought from polluted air or lifestyles with heavy smoking, coincide with regions of more cases, such as Italy, America, and China. Specifically, the aforementioned high-risk groups have been increasing in prevalence, with an aging population and high percentages of obesity. For example, in America alone, over 34% of adults are defined as obese. Previously, viral diagnosis has been achieved through a variety of biochemical and molecular techniques, with some approaches specific to certain viruses. In the case of RNA virions, and in particular COVID-19, where the option of capable tests were bare, reverse transcriptase polymerase chain reaction, known as RT-PCR, has already proven useful in identifying asymptomatic cases of the virus and acting as pathogenic confirmation in symptomatic patients. COVID-19 clinical presentations are highly varied, characteristically analogous to food poisoning cases where general enteric systemic misfunction is prevalent. Specifically, the novel coronavirus-related symptoms are considerably less conservative, spanning from innate immune response-associated fever to anorexia and melligius, among other deadly complications such as pneumonia. Furthermore, Udagama et al. Established that regardless of respiratory symptom presentation, diagnostic differentiation from non-specific symptoms of COVID-19 etiology and other pathologies are inaccurate, and the usage of molecular techniques offer greater specificity due to the selective primers involved. However, the tools utilized in COVID-19 diagnosis, such as the aforementioned RT-PCR, have already faced shortages of the unique primers required for sample preparation. In locations that required immediate pathology identification, such as Wuhan Central Hospital, diagnosticians switched to computerized tomography scans as an alternative, potentially misdiagnosing patients with other pneumonia-complicated viruses. This was due to their strong sensitivity at 86 to 98%, or low specificity at 25%. The lack of consistent symptom presentation, coupled with an infectious asymptomatic period capable of lasting weeks, alongside worldwide shortages of necessary primers for COVID-19 RT-PCR and the ongoing pandemic's strong infectivity potential, which expressed an R0 value of 2.2, necessitates the introduction of rapid and highly specific in situ diagnostic kits. Long et al. Utilized biochemical serological testing to determine antibody responses from COVID-19 virions to aid in the diagnosis and in some cases prognosis of infected patients. Specifically, Long et al. Aimed to adopt antibody seroconversion from antibody, immunoglobulin G, and immunoglobulin M COVID-19 affected patients to quantitatively measure and further affirm COVID-19 pathology outcome, with Figure 3 shown as an example, which until a vaccine is both created and assessed as viable, may present another diagnostic facet for COVID-19 prevention and transmission control.
In total, 285 COVID-19 patients were enrolled from 3 hospitals in the province of Chongqing, which is located adjacent to the Hubei province from which the outbreak started. All 285 enrolled patients were confirmed to be infected with COVID-19 via RT-PCR assays for nasal and pharyngeal swab specimens. In addition to the initial study, a follow-up study was performed using the serum of 63 patients at Yongchuan Hospital. In order to evaluate the effectiveness of the serological test, 52 patients with suspected COVID-19 and had failed RT-PCR results were enrolled from Guangzhou People's Hospital. A further serological test was performed in order to identify asymptomatic infections in close contacts, of which 164 contacts were identified. Analyses of IgG and IgM levels against COVID-19 were performed on patient samples that were inactivated at 56 degrees for 30 minutes and stored at -20 degrees Celsius before the commencement of testing. These samples were tested using a magnetic chemiluminescence enzyme immunoassay, which was developed based on a double antibody sandwich immunoassay. In a double antibody assay, a capture and detection antibody are used to detect different epitopes. The capture antibody binds to the antigen, and the detection antibody binds to the capture antibody. The immunoassay in this experiment was conducted on an automatic magnetic chemiluminescence analyzer, with alkaline phosphatase conjugated IgG/IgM antibodies being used as the detection antibody. Statistical analyses on these studies were expressed as an IQR and were compared using a Mann-Whitney U test, where 2 independent groups are examined with the dependable variable being either ordinal or continuous, but not normally distributed. Categorical variables were expressed as a numbered percentage and were compared using Fisher's exact test, which determined if there were any non-random associations between 2 categorical variables. A p-value of less than 0.05 was considered statistically significant. The analysis software used was R software. In the study of the cross-section cohort of 285 cases, antibody detection rate based on the number of days after the onset of symptoms were determined. Patients were grouped based on symptom onset days, where the levels of IgG and IgM that were detected in the cohort gradually increased with days after symptom onset. The positive rate of IgG reached 100% at around 19 days after symptom onset, whereas IgM reached a peak of 94.1% around 22 days after symptom onset. In the follow-up study, collectively from the 63 patients, a total of 281 serum samples were collected and tested for IgG and IgM against SARS-CoV-2. These samples achieved an overall seroconversion rate of 96.8%. All patients achieved a seroconversion of IgG and IgM within 20 days after symptom onset. In order to evaluate the effectiveness of serological tests in a COVID-19 diagnosis, 52 COVID-19 suspects with negative RT-PCR results were enrolled. Of the 52 cases, 4 patients showed positive IgG or IgM in their first samples. Patient 1 had an increase in titer of IgM in sequential samples. Patient 2 had positive indications for both IgG and IgM. Patient 3 showed a 4-fold increase in IgG towards the end of the study, and patient 4 had an extremely high level of IgG and IgM titers, which was an increase of 110-fold more than the cutoff values. Finally, in order to identify any potential asymptomatic patients, further IgG and IgM serological assays were performed. 164 close contacts of a couple with a confirmed case of COVID-19 were closely monitored. Of the 164 individuals, 16 of those close contacts were confirmed to have COVID-19. This was confirmed via RT-PCR tests. The remaining 148 individuals showed no symptoms and had a negative PT-PCR result. Following the results of the serological assay, 7 individuals were confirmed to have increased levels of IgG and IgM, suggesting the presence of COVID-19.
The sudden emergence of the novel coronavirus, coupled with its multiplicity of deleterious outcomes and efficacy of transmission, has necessitated the sophistication of clinical biochemical techniques to corroborate preliminary diagnosis. Patient serostatus, due to the innate immune response to pathogenic stimuli, is a historical determinant of infection, and the diagnostic significance of serum antibody testing using real-time PCR cannot be dismissed. Long et al. Delineate a positive increase in immunoglobulin G and M antibodies correlating with symptom onset. As seen in Figure 5, immunoglobulin G and M reached positive rates of 100% and 94.1%, plateauing at 19 and 22 days respectively. The elevation of antibodies had a statistically significant correlation with disease severity, indicating its potentiality as COVID-19 infection and severity index biomarkers. The immunoglobulin staggered kinetic profile is corroborated by Paduan et al. And Jin et al., who observed analogous trends in antibody-positive rate kinetics, as seen in Figure 9 and 10. These findings indicate conclusive exclusion of COVID-19 diagnosis if antibody profiles are undetectable 20 days subsequent to symptom onset. The longitudinal seroconversion profiles of immunoglobulin G and M antibodies show 100% of patients achieved seroconversion at a median of 13 days. Similar to SARS and MERS, there is no statistically significant chronological order of seroconversion, supporting the simultaneous testing of immunoglobulin seroconversion in COVID-19 diagnosis. The confirmatory criterion of MERS, which has formulated the basis of COVID-19 diagnosis, suggests seroconversion or a greater than 4-fold increase in immunoglobulin G. Long et al. Suggests this criterion may be too resolute to confirm some COVID-19 patients. As evidenced in Figure 11, 29% of COVID-19 confirmed patients did not fit this criteria and may account for false negative results. Figure 12 shows the heterogeneous analytical sensitivity of real-time PCR, limited by biological source, sample collection, and detection limits. To evaluate the efficacy of antibody response serological testing, Long et al. Examined suspected COVID-19 patients who returned negative real-time PCR results. As seen in Figure 7, 4 cases demonstrated positive immunoglobulin G or M presence, substantiating COVID-19 diagnosis, suggesting serological testing may mitigate the limitations of and offer potential alternatives to real-time PCR. This study corroborates the hypothesis of Long et al. On the efficacy of antibody testing in the diagnosis and epidemic survey of symptomatic and asymptomatic COVID-19 infections. The Ethics Commission approved multi-center cross-sectional study design and single-center follow-up study composition allow Long et al. To gain a holistic interpretation of antibody diagnostic testing. Long et al. Delineate the study was limited by singular case serum sampling, and despite attempts to diversify population demographics, it is limited to the Chongqing municipality, and thus cannot be used in isolation to make broader generalizations and only when viewed in conjunction with larger cohort studies. The application of CFDA-approved reagent kits, use of Exceed 260 automated magnetic chemiluminescent analyzer, and statistical analysis using R software version 3.6.0 attenuated potential sources of error, bolstering reliability and accuracy of clinically significant data. The methodology and results are clinically applicable to industry practice and emulated in the aforementioned works of Padawan et al. And Jin et al. The implementation of longitudinal immunoglobulin profiles demarked an early immunoglobulin G seroconversion time as the leading cause for false negative diagnosis at 12.2%. 2 patients displaying delayed seroconversion time were lost due to undisclosed reasons in follow-up observations, and the inclusion of this data must be taken into consideration when reviewing seroconversion profiles as a confirmational criterion of COVID-19 infection. Potentially relevant conflicts of interest and financial support were disclosed, authenticating author transparency, however, should be taken into consideration when reviewing publication bias. Clinical biochemical techniques are the nexus for pathological diagnosis. The principal armamentarium of coronavirus etiological diagnosis has historically been the molecular biochemical identification of viral RNA in respiratory tract specimens due to the variability of real-time PCR. The amalgamated works of Long et al. And others indicate the potential validity of alternative biological matrices in COVID-19 diagnosis. As real-time PCR techniques require stable power supplies, complex apparatuses, and several hours for a quantifiable outcome, the future of COVID-19 diagnostics will inevitably resolve rapid and easy-to-use detection methods. Zhu et al. Demonstrate the efficacy for loop-mediated isothermal amplification coupled with nanoparticle biosensory assays to facilitate rapid diagnostics in field laboratory environments where resource and infrastructure limitations are commonplace. As seen in the Figure 13 workflow, the diagnostic process from sample collection to interpretation takes 1 hour. The simultaneous amplification of open reading frame and nucleoprotein primer sets had a sensitivity of 12 copies per reaction, with no cross-reactivity. There was an analytical sensitivity and specificity of 100% in oropharynx specimens. The synthesis of LAMP amplification, reverse transcription, and nanoparticle biosensory multiplex analysis offers an economic and time-resourceful COVID-19 diagnosis to facilitate timely therapeutics and infection control. Due to the elevated COVID-19 associated mortality in older demographics, alongside those with underlying medical conditions, and the heightened transmission risk of collecting oropharyngeal specimens, non-invasive diagnostic techniques are of critical importance to maintain quality of life. Building on the research of alternative biological matrices by Long et al., retrospective analysis on the diagnostic relevance of saliva samples are being conducted. Kah et al. Demonstrate COVID-19 virus detection in saliva by viral culture, and 91.7% of self-collected samples in confirmed cases had a positive COVID-19 detection. As displayed in Figure 16, Kah et al. Presented saliva as a medium for serial viral load monitoring, as there was an observable correlation between therapeutic intervention and declining viral load, thus substantiating the utility of saliva samples in COVID-19 clinical investigations. The immediate pervasiveness of the COVID-19 pandemic has placed an exorbitant strain on many facets of the globe. We can't even present this assignment in person, as initially intended. The significance of immunoglobulin G and M antibody response in serological diagnosis of COVID-19 is crucial to intervention diagnostics to attenuate progressive transmission and secondary complications associated with respiratory centralized pathogens. The work of Long et al., coupled with the exploration of non-traditional specimens and techniques, further the collective understanding of COVID-19 in an attempt to reduce the impacts of the current novel coronavirus, so we may be more prepared for the next one.